Hello everyone, today we'll talk about the medicinal chemistry of protein synthesis inhibitors. Let's start with the objectives, but as you know, any comments I say here is just for your own sake, your own education. Basically, you'll only be responsible for the notes, okay? So, objectives, you should be able to describe the protein synthesis process. We'll go through the two stages, transcription, translation, and the steps of each one. So just to get familiar and understand the protein synthesis process, such that when we refer to these steps, how we inhibit them with the classes of anti microbial agents, then you will understand what we are talking about. So then you need to compare between these different mechanisms. I mean, not compare, but at least recognize the difference between these different classes of protein synthesis inhibitors. And as far as the structures, the only one really I want you to pay attention to is the tetracyclines, which is very easy. If you see a structure with 4 rings, then that must be a tetracycline. The others, just be familiar with how the structures look like, just the general structure. And then I hope you'll be able to compare also the structures you will see today with the ones you've seen in the previous class, the beta-lactams. Okay, and then describe the techniques used to overcome the acid instability problem with erythromycin, as you will see very soon. And then appreciate the importance of the use of esters in designing prodrugs to enhance several features of drugs, such as palatability, basically the taste, and the solubility and stability of drugs. So in this this lecture, you would see many examples of prodrugs using ester to enhance certain features. Okay. Let's start by an overview to the protein synthesis process. As you might know, proteins are very essential to controlling the cell activities, such as catalyzing reactions. As you know, enzymes that catalyze reactions are actually protein in nature. And they also can do functions like transportation, and they're responsible for the growth of the cell as well as replication. And proteins, they don't exist forever. They are usually degraded or exported out of the cell. So the protein synthesis is an important process for the cell. And the protein synthesis goes through two stages. The first one is called transcription, following these steps, and translation following these steps. So we are actually gonna look at a quick video here just to give us an overview of the protein synthesis.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anti-codon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these three parts will make translation happen. Translation occurs in the cytoplasm of the cell, wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then, the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first three letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column, and the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis.
So now that we have a good idea about the protein synthesis, how can we inhibit the synthesis of protein, especially for bacteria, in order to cause the death of the bacteria or other microbes? We could target the stage at the transcription. In this case, we target the nucleic acid replication or the making a copy of the mRNA, and this is something we'll cover later on, not at this lecture. But today we'll talk about antibiotics that targets the translation process. Okay, so in this figure here, there are several types of antibiotics, and they target different stages of the protein synthesis process. And luckily, the ribosome for the bacteria is different from the ribosomal units in the human, so this helps with the selective toxicity. Plus, there's another factor, which is the diffusion rate through the barriers to the bacterial cells are much faster than those to the mammalian cells. So that's another factor that also helps with the selective toxicity. This figure here shows the different types of antimicrobial agents that can inhibit the protein synthesis. Some work on the 50S subunits, others work on blocking the transfer RNA from binding to its sites. Others, like chloramphenicol, blocks the peptide transfer, basically blocks the making the peptide bond between the amino acids. And these antibiotics, the macrolides and the aminoglycosides, they block the translocation step. Let's start with the tetracyclines. So tetracyclines bind to the 30S subunit and prevent the binding of the transfer RNA. Therefore, the growth as well as the release of the protein is inhibited. And notice that in the many of these protein synthesis inhibitors, they only stop the growth of the bacteria, but they do not kill it. So it's a bacteriostatic, and so they rely on the human antibodies to attack these bacteria and get rid of them. A factor that plays role in the selective toxicity in the case of the tetracyclines is that the diffusion rate for the bacteria is much faster and higher than in human. And the tetracycline will pass through both the outer membrane through the porins through a passive diffusion, and it will pass through the inner membrane by a carrier protein. So this is an active process, this is a passive process. Therefore, they can affect both the gram-positive and gram-negative bacteria. Example on tetracyclines are tetracycline, doxycycline, minocycline, and diacycline, which this last one has poor oral availability, so it's typically given in IV infusion only. Notice that in the case of the tetracyclines and all protein synthesis inhibitors antibiotic, they really have to pass through not just the outer membrane of the bacteria, but also the inner membrane in order to reach the cytoplasm, which is where the synthesis occur. This is different from what we have seen in beta-lactams, where they only had to cross the outer membrane in the gram-negative, and of course with the gram-positive they didn't have an outer membrane. You might remember this from previous session, which is tetracycline form chelation complexes with multivalent, basically di and trivalent metal ions such as calcium, magnesium, aluminum, as well as zinc and iron and bismuth. These agents, you can actually find them, like in the case of calcium, they're available in the cheese products like milk and cheese, and the magnesium, aluminum, you can find them in the antacids. Bismuth, of course, in the case of pepto-bismol, and iron in the iron supplements, zinc in the multivitamins. So you should avoid administering all these agents along with when you take tetracycline. Otherwise, a chelation complex will form, and it will reduce the absorption of the tetracycline, and it will reduce the antimicrobial activity of the tetracycline. You should separate the tetracycline from any of these products by at least 2 hours. And I hope you still remember in the case of bismuth, if you start with the bismuth first, then you have to wait 6 hours before you can administer the tetracycline. Okay. An important side effect for tetracycline that you need to pay attention to is that the calcium-rich structures that are being developed, such as bones in children and teeth in children as well, the tetracycline binds to these calcium-rich structures, and they form a permanent yellow or brown in advanced cases discoloration. They might also weaken the bone in extreme cases when the tetracyclines are administered on higher doses for a longer amount of time. So that's why they should be avoided in children and in pregnant women. Another side effect for tetracycline is the phototoxicity or hypersensitivity for certain tetracyclines, and in this case we'll advise the patient to avoid the sun at least during the first few doses, and maybe use some sunblocks. Macrolides are usually used as the second choice after penicillin in case of patients who are allergic to penicillins. Drug interactions with macrolides is actually very common because they are inhibitors of the cytochrome P450 3A4, and therefore they will increase the concentration, the plasma concentration for substrates for these enzymes. For example, the statins, if they are co-administered with macrolides, then the metabolism of statins will be inhibited, This will result into increased concentration of the statins, and then you will see more of the side effects of the statins. The most common one is the rhabdomyolysis, which is muscle pain. The first example on macrolides is erythromycin, which is a 14-membered macrocycle lactone with sugar and amino sugar attached. There are two sugar units here. And erythromycin is unstable to the stomach acid because of the presence of this lactone here next to these OH group, this one here and this one here. X in the erythromycin would be an OH. So at acidic condition, these will reform to result in a ketal cyclic internal structure, ketal cyclic, that is actually unstable, and this is believed to cause, or its degradation products are believed to cause the GI cramping. So the erythromycin is not considered stable into acidic conditions, but there are some solutions for this problem here. So the first solution would be using an enteric coating, also known as delayed release. I hope you still remember this from the proton pump inhibitors. So with the delayed release or the enteric coating, we could protect the erythromycin to pass through the stomach acid to the intestine, and then where it gets absorbed. Another way is to actually protect or mask this OH group here with a methyl group. So instead of OH would be O-methyl, and this will result into a new molecule called erythromycin, which we will see here. That is actually acid stable, and therefore it could be used for treating ulcer cases caused by Helicobacter pylori, which has and erythromycin also has greater activity than erythromycin for that reason. And I mean because of the different modification in the structure here, it ended up having better activity. Anyway, so and the third way of solving the acid stability is by increasing the size of the macromolecule to 16 ring. Basically, these would be separated from each other, and then in this case they will not be able to form this ketal cyclic, and this is actually remind us of the azithromycin, which is, although it is not 16 but it's 15 membered ring with a different structure that does not allow for this ketal internal cyclic reaction to occur. So it is acid stable, and you might imagine that it can also be used for Helicobacter pylori, although there is not enough studies to show that. But I just looked at an article in 2012 that compares the effect of azithromycin and clarithromycin on the Helicobacter pylori, and they both have the same results. So they can actually be used based on that article. And what's unique about azithromycin, is that it has a long half-life. So because of its longer distribution to the tissue, so this allows the once daily dosage. You probably have seen the Z-Pack in your pharmacy, where you have 2 tablets as a loading dose at the first day, and then one for 4 days, so a total of 6 tablets. So this allows once daily administration, which is very convenient, and as you should know by now, conveniency is always good to help patients comply. Okay, so just different techniques to solve the acid stability problems. Another problem with erythromycin that it has an unpleasant taste, which is actually common with macrolides. And to overcome this problem, there are certain ways. I'm going to start with the easier ones. So you could actually use a film coated, like sugar coated tablets, just to cover that taste until you swallow the tablet. or you could use the enteric coating, which would do the same thing and would serve two purposes. So the enteric coat, it would serve the purpose of protecting the erythromycin from the acid and also mask the unpleasant taste. And the other ways to take care of the unpleasant taste are by designing a prodrug such as erythromycin ethyl succinate. So by assigning an ester group to the erythromycin at this location here, next to the OH here, we remove the OH and put an ester group. This can be used to mask the taste, the unpleasant taste for the erythromycin. And this formulation, the EES, is typically used to formulate oral suspensions for pediatric use. In fact, if I remember correctly, the EES is also available in tablet forms, not only in suspension. But don't quote me on that. I can't remember it now. And then another way is to form an insoluble salt. So this is erythromycin stearate, which is an insoluble salt. It's not a prodrug. It's a salt that is insoluble. So by making the drug insoluble, then it's not gonna dissolve in the mouth, so you're not gonna be able to taste the unpleasant taste. I just wanted to compare between, make sure you understand the difference between film coated versus enteric coated. The film coated could be coated with a sugar, for example, just to allow the drug to pass through the mouth and avoid the bitter taste, while the enteric coated is coated with a material that is only degraded into the small intestine, and it would protect the drug from the acid. In this case, the enteric coated can also be used to protect the drug, to mask the unpleasant taste. Finally, to enhance the water solubility of the erythromycin for injections, there's a dosage form called erythromycin lactobionate, which would make the erythromycin more soluble. I just want to comment on one thing here, which is the use of the prodrug, in particular the use of the ester in this case, to mask the bitter taste. I hope by now you have noticed that we use this technique, which is adding an ester group and designing a prodrug to solve many problems. For example, we could improve the solubility of the drug by adding a small hydrophilic ester group. We could, on the other hand, if we need to improve the lipophilicity of a drug, we could add a long chain of ester as an ester group to the drug, such that the drug becomes more lipophilic. So this way of designing the drugs is very common, and we rely on the esterase enzymes to convert this prodrug to the active form. Okay, so we have seen some examples, but this is the first time where we are seeing a prodrug using ester group to mask the bitter taste. The next class of antibiotics that inhibit protein synthesis is the lincosamides. And although they are structurally different from macrolides, they actually have a lot of pharmacological similarities to the macrolides. Similar activity and mechanism of action, they bind to the 50S ribosomal subunit and inhibit the translocation, and therefore they cannot be combined together with the macrolides, because again they compete on the same side and they cancel each other's effect. Okay, clindamycin is an example of that. It can be taken orally for bone and joint infection and topically to treat acne. And the same problem with the macrolides about the bitter taste, the unpleasant taste. You could formulate the clindamycin into a water insoluble palmitate prodrug, such that it can cover the unpleasant taste. Is also a form that's available orally. And just like the macrolides also, clindamycin can be used for people who are allergic to penicillin. So in case you have to choose, in case you cannot give penicillin, then these are one of the alternatives besides the macrolides. Okay. The next class is the oxazolidinones. They inhibit the protein synthesis at a much earlier stage before the 50S subunit binds to the 30S subunit. So they inhibit that binding, and again, it's about chemostatic. The unique thing about the oxazolidinones um, is that it actually has a broad spectrum of activity that covers even the MSRA. Okay, examples are the linezolid, brand name for it is Divox, and the TDzolid, the brand name for it is the Civextro. There is one side effect for the linezolid, which is it inhibits the monoamine oxidase. So patients should be careful if they are taking a monoamine oxidase inhibitor drugs. These are example of these drugs are the antidepressants. Also, they should be careful if they are taking any adrenergic, serotonergic drugs, because again, the monoamine oxidase inhibitors would increase the serotonergic and adrenergic effect. And they should avoid eating tyramine containing foods, because tyramine is metabolized by the monoamine oxidase enzyme. So if we inhibit the enzyme, tyramine concentration will increase, and then you will have headache and problems, side effects from the tyramine. Let's see, the TID solid was approved in 2014, which is more potent and can be administered once a day and has no monoamine oxidase effect, so it's actually advantageous over the linezolid. Okay. Like I said in the previous lecture about in the beta-lactam antibiotics, please pay attention to the brand names. Make sure you memorize the brand name next to the generic name, although most likely I will provide you with the brand name in the question in the exam, but I hate for you to miss a question if you don't know the brand name for the drug. Okay. The next class of antibiotics that inhibit the protein synthesis is called the streptogramines. Examples are the combination of these two products together in a drug called the two agents called quinopristine and dalfopristine under a brand name called Sinercid, which is a mixture of these two agents. They bind to the 50S but at different regions. The quinopristine prevents the elongation, while the dalfopristine prevents the transfer of the peptide chain. So, from the tRNA to the next. So when they both work together, they provide a synergy effect. That's probably why the name Sinercid. And although each one is bacteriostatic, but when they are combined together, it can kill the bacteria, and it becomes bactericidal. And these are typically reserved for the life-threatening gram-positive infections when other antibiotics fail. Next is chloramphenicol, which binds to the 50S subunit, inhibiting the movement of ribosome along the messenger RNA, so they inhibit the transpeptide, the peptide chain transfer. It's also bacteriostatic. And like we said with the macrolides and the glycosamides, they should not be combined with chloramphenicol because they bind to the same sites and they compete with each other, they cancel each other. The structure of the chloramphenicol here has a unique function group here called the nitro, and it causes toxicity. It can be actually responsible for some blood diseases such as the pancytopenia. In fact, many cases, whenever you see a nitro group attached to a phenyl group, there is a chance that there is a toxicity. Just for your information. Um, Also, in babies, because this drug is metabolized through the glucuronic acid, when this step is not completely mature in babies, so this drug is not quietly metabolized, so this can cause toxic effects such as gray baby syndrome, which could be fatal. So this drug should be avoided in babies. It's widely used for eye infection and could be used for infection as well. Can have hypersensitivity reaction in about 10% of the people. Again, prodrugs. In this time, products are designed to improve the water solubility, and of course, in this case, the drug is administered IV. So if you want to administer the drug intravenous, then you have to improve the solubility of the chloramphenicol. A prodrug called chloramphenicol hemisuccinol ester is designed, and then it actually disintegrates when it reaches the blood by the esterase enzymes. The thing is, it's not given intramuscularly because it cleaves very slowly, and it's not good for antibiotic to be given slowly. We want it to be working immediately at higher concentrations, so it's better to be administered intravenously. But since we mentioned that there are many drugs where we want them to be released slowly, so we formulate them in a prodrug form and ester form, and then administer them by intramuscular injection, and they are released slowly, and that could be to our advantage. But this is not the case with the chloramphenicol. And the last class of antibiotics that work by inhibiting protein synthesis is aminoglycosides. We will visit this class again. I just thought since it belongs to this class, this mechanism of action of inhibiting the proteins, I thought it would be good to mention it along with the other classes. So this is the structure of the streptomycin and gentamicin, examples of aminoglycosides. They bind to the 30S ribosomal subunit and prevent the movement of the ribosome along the messenger RNA, so they inhibit the translocation. So the triple code cannot be read. They are bactericidal, not static, because of their effects on both the ribosomes as well as on the outer cell membrane of the gram-negative. So they actually affect the outer cell membrane, the penetration of because they affect the way molecules penetrating the outer cell membrane, and this disturbs the cell. So in addition to their effect on the ribosome, so this effect when combined makes the aminoglycosides bactericidal. Okay.